I'm recording today, so we'll have this recorded if you need it. We're going to be on Module 11 this week, looking at the circulatory system. Next week, when we get together, we will dissect the heart, all right, and do a couple other things. And the week after that is spring break already. How about that? That just seems like it came really quick. So then it works out well for us that we'll have the lab. You don't have to study over spring break, basically, unless you want to start reading Module 12, okay? So then when we do get back on Module 12, we'll be doing the lecture and the lab on the same day. That's how we're going to catch up from our ICE day. Question?
What's the difference between a circulatory system and the respiratory system? I thought they kind of work together.
They do work together,
Okay.
yeah. But when we get to the respiratory system, which is Module 14, we get into the details of cellular respiration. Okay, we don't get in those details today. Okay, so that's why I consider Module 14, respiratory system, the most difficult of all the modules. Okay? Circulatory is a lot of anatomy versus physiology, right? So we have a lot of stuff we can see with our eyes, which is very cool. All right. So go ahead and open your lab books to Module 11A. I don't know what the lab number is. What's the lab number? 22, lab 22.
Page 78.
Page 78. All right, so we're going to be looking at the whole circulatory system throughout this module, and it's going to include all of these things. We'll be talking about blood, hemopoiesis, which means the manufacture of blood, right? Where does that take place?
So,
In the bones, right? Hemostasis is how we keep blood the right consistency it needs to be. In other words, it's going to clot when it needs to clot, but it's not going to clot unless it needs to clot. That kind of thing, okay? Keeping it all even. Kind of like homeostasis, but hemo meaning blood, keeping the blood at the constant level. That also refers to pH and volume, like how much water, that kind of thing in there. We'll talk about blood typing today, although we're not going to actually do blood typing until we do the next module. And the reason we'll do that is because the next module is about immunology. And so really blood typing is all about how we can find out what our type is by checking the surface antigens, okay? So that's why I moved that whole thing. It might be in your book. Your textbook might talk about doing your blood type yourself. We're moving that all the way to the next one, so the week after spring break. But we have a lecture that day too. All right. You can just leave early. Okay. All right. Circulation. It's called the circulatory system for a reason. There's a pump that circulates everything through your body. The pump is called the heart. We have something known as cardiac cycle. Dum, 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 dum. All right, and there's a thing that causes all of that. Blood pressure and blood vessels. So we'll be covering all of these things throughout here. So speaking about the circulation part, about when you look at here, I have a figure 8 on here. So really what's happening is our heart is like a double pump. And we end up pumping things up to the brain, and then down to the body, and up to the brain. And as it goes from here to there, the heart's gonna be pumping, but it has to also go to the lungs where it can be oxygenated. Okay, it's all gonna work together there. All right, let's start with blood. What are the functions of blood? Do y'all remember what type of tissue blood is? Think way back. Type of tissues. There's only 4 different types of tissue.
it's not muscular.
It's connective tissue, right? It's connective tissue, and you might think that's kind of weird that blood would be connective tissue, but think about it. Blood clots, doesn't it? And it's going to connect things. When you have a cut, that blood's going to come and kind of mend things through the clotting process. All right, so but the first function of blood is going to be transport. Obviously, it's like a highway taking all of the things we need in our body to the places our body needs them. We'll be taking oxygen to the tissues. We'll be getting rid of CO2, which is a byproduct and a toxin. We're getting rid of that by breathing it out. And fortunately, that toxin to us is a nutrient to plants, right? So CO2 is good for them. And so we live in this beautiful symbiotic relationship with them. We also take nutrients from our intestines that got there because we ate food, got absorbed in the intestines, and then took that to the cells that need it. And the one thing in particular that our cells want from our food is what? Glucose. Did somebody say glucose?
No, I say ATP.
But that's so we can make ATP. That was the next question. Yeah. We also make wastes during any kind of cellular activity. The wastes go to the kidneys. They also go to the large intestine. And then, of course, hormone transport. We learned that from the last module, that the way the hormones get to the target cell is through the bloodstream. All right, so all that's being transported by the blood. Number 2, regulation. Keeping our body regulated. Homeostasis right there. That's the volume of our fluid. There's a certain amount of volume we want. We don't want our blood to be sluggish. All right. Temperature equalization. When you get hot and you're trying to cool off, you turn real red because your blood is coming to the surface to help cool you off. And then pH levels. It turns out the pH level is very tight range of pH level for your blood. And 3, protection against fluid loss. Biohemostasis. Here's our hemostasis, is keeping that volume right by going through the process of blood clotting.
What is I know what that means in physics, but in
Which one, hon?
on regulation, equalization.
Oh, delta T?
Yeah.
Change in.
Change in.
Temperature. Delta just means change in. Sorry about that. Fluid loss via hemostasis, blood clotting. Fighting infection, because the blood not only carries oxygen and nutrients, it carries white blood cells. And white blood cells are there, part of our immune system. We'll be looking at the types of white cells today, but we're going to study them in more detail. When I have you take a blood sample from yourself, we're also going to make what is known as a diff slide, and I'll get to that. Diff slide means differential. And so hopefully, if everything goes right, and we have a decent slide, we'll be able to see the types of white blood cells you have in your body. If anybody wants to be a little sick that day, you'll have more white cells. We'll see them under the scope. Go ahead and come on in. All right. Oh, there's our blood coming down. Whoosh. Okay, so blood. All right. Characteristics of blood. Can y'all see that? I'm sorry, I'm being too fancy with my red print right there. Viscosity. Viscosity is the resistance to flow. Do you have these words written down, like viscosity? What do you have?
Yeah.
Right. How thick is it? How thick is the blood? That's what viscosity means. It says here alteration of shape due to cohesion. In other words, when we're talking about the red blood cells, they can be bent into shape very easily unless there's a condition that we'll talk about. Plasma, I need to change these letters to white, yeah? Plasma, the fluid portion of blood, mostly water. Formed elements or solid, that's the solid portion of blood, known as RBC. RBC stands for red blood cell, also known as erythrocytes. That's the technical name for them. Where did my pen go? Red blood cells. Also known as erythrocytes. So we have the plasma, which is the liquid portion of the blood. The formed elements are the solid portion of the blood. WBC stands for white blood cell. And also known as leukocytes. Means, eryth means red, and leuko means white. Platelets. Platelets are cell fragments. They help with clotting. Platelets are kind of weird. They're not really whole cells. They're just fragments. And they, at certain times, basically when they're touched by air, they get sticky and they swell up. And when they get sticky and swell up, they will help blood to clot. It's crucial, though, that we don't have any blood clotting taking place when it shouldn't be. Okay, in fact, some of the most common, terrible occurrences out there, things like a heart attack or a stroke, both of those are typically caused by blood clots in the wrong place. Clots that travel to your heart, blocked off part of your heart attack, called a myocardial infarction is the official name for that. And then a stroke, of course, when we have a ischemic attack.
So what can be the causes of blood clotting?
For some reason, you're making clots. For some reason, there could be. Typically, what will happen is if the inside of your blood vessel is rough, and it should be nice and smooth. How does it get rough? Things get deposited on it, like cholesterol, and it makes a little kind of sharp edge, if you will. And when the platelets see a sharp edge, they're like, oh no, I gotta make a clot, So they get confused, if you will. Can everybody see these okay? I apologize.
When it needs to clot, like if you slice your hand or something, it takes cholesterol from somewhere else, or does it use
No,
what it has?
cholesterol happens to be making it sharp, right? That's making the inside of the vessel, I'm using the word sharp, it makes it irregular.
So it like grabs together and that's what makes it clot?
Yeah, so in other words, the platelet doesn't care what it is, it makes it look irregular. In that particular case, it tends to be cholesterol. You need to understand, cholesterol is very good for you. Cholesterol is good. It's just if it's in the wrong place, just like anything, right? Then it can cause a problem. But when you cut yourself, we'll get to that in a minute. When you cut yourself, that opening like there creates a sharp edge, and the platelets, boom, they go in there and they swell up and they collect up on there. So the three types of blood cells, even though thrombocyte, which is a platelet, isn't really a cell. Platelet. Thrombo means to clot. So we have erythrocytes, leukocytes, and thrombocytes. The erythrocytes are the red blood cells, the leukocytes are the white blood cells, thrombocytes are the platelets. And these are all three part of what are known as the formed elements of the blood, the solid portion of blood. Typically they'll show the leukocyte, the white blood cell, being quite a bit larger than the red blood cell, which is actually the case. Most of them. There's one small white blood cell, the special one.
So it's red, white,
Red what?
red blood cell, white blood cell.
Red blood cell is erythrocyte, leukocyte's a white blood cell, thrombocyte's a platelet. Okay? So if I ask you what are the types of blood cells and you tell me RBC, WBC, and platelet, that will be the correct answer. If you tell me erythrocyte, leukocyte, and thrombocyte, that will also be the correct answer. Okay?
Is that a hint for this quiz?
Probably, I don't know. Okay, so this is a tube right here, this test tube that's showing you. This is what I used to do at the vet clinic all the time, is I would get a blood sample and put it in something called a clot tube. And what would happen is you put it in the tube and it would clot, it would literally clot. And then you take the whole tube and put it into a centrifuge, which would spin it around. And all the formed elements, all the solid portions would go to the bottom of the tube like this. And then I would have all this plasma at the top. And why did I want? I wanted to test the plasma for various things. You know, for liver enzymes, for, you know, whatever I was testing it for. Also, you would see a little tiny line of white. And those are literally the white blood cells that form a little tiny line right between here. Sometimes if I had a really fat line in there, guess what that told me?
You're sick.
They had a high white count, and they're sick from something. They were producing a lot of white cells. And so I'd know that. I would also do a white blood cell count with a machine. Actually, I could do it on a slide. That was the old-fashioned way, and then we had a machine. So blood is viscous and dense. That means it's thick and it's heavy. The red blood cells actually literally have metal in them. They have a metal called iron in them. You probably know that. Someone who's low on iron would be anemic. To be anemic means your red blood cells don't have as much iron as they could. All right, it has a pH of between 7.35 and 7.45. Wow, that's a tight range right there. We don't want it to get any lower, which would be more acidic, or any higher, which would be more basic. And we have about 5 liters for an average adult. You know, it could be a really big guy, could have more than that or whatever. 55% of it is plasma or liquid portion. And of the liquid portion, 91% of that is water. Then we have proteins, blood gases, ions, wastes. A lot of times we would take blood gases there to find out how much, you know, CO2 is in there, are our kidneys functioning, are our lungs functioning well to get rid of it, this kind of thing. Proteins including albumin. Interestingly, albumin is the same stuff you find in the white of an egg. That kind of icky stuff. We have that too. That helps with water regulation. We have something called globulin, fibrinogen. Those are to be recruited if clotting is needed. And 45% of the formed elements, 99% of those are RBCs, 1% are WBCs, and platelets together. So that's why that little line is so thin, because we don't need as many white blood cells. We only need white blood cells at certain times when we have to fight disease. There's always white blood cells like patrolling, if you will. They're like the cops of our body, right? They're patrolling and they're finding if there's any issue that needs to be dealt with. And if there is, if they come across a bacteria that needs to be gobbled up, they'll send a message to the bones to say, make some more white cells. And then a particularly special white cell actually can be manufactured somewhere besides the bones. It's pretty cool. All right, let's talk about the red blood cells. They have a unique shape. They are anucleate. You know what that means? They do not have a nucleus. Red blood cells do not have a nucleus. So what can they not do? Can't reproduce. They can't reproduce, okay? Because here they have one job, transport. That's what they're going to do. They're going to be transporting oxygen in that little heme, that little iron thing in there. So they kind of have a donut shape. They don't have a hole in the middle. They just have a thinness in the middle. So in a microscope, they kind of look like a donut. They have no DNA. They do not go through mitosis. They have a short lifespan of about 4 months, which is pretty long, actually. And guess what's constantly being made all the time in your bones? Red blood cells. Boom. I think it might tell us here. Main job to carry oxygen on the hemoglobin. Continuously made in the bone marrow. Look at there, 2 million per second. Whoa, our bones are making 2 million RBCs per second because we really need them. Pretty crazy. So the ones that are being replaced, that must mean per second about 2 million are dying. A low number of RBCs or hemoglobin, which is the protein that's holding the iron. So a low hemoglobin is called anemia. It's measured with a test called an H&H, hematocrit and hemoglobin. And that tells you, once again, we put the blood in a tube, little tiny glass straw. I have some of those, I'll show y'all. And we spin that down in a special centrifuge, and we can see where that red line is. And if the red line is not high enough, we know that they are anemic. The H&H is 44 to 48 for males, 38 to 42 for females. And if it's lower than that, they're anemic. If it's higher than that, they're probably dehydrated, and they need more water in their system. There is a condition called sickle cell anemia. That's a genetic deformation of the red blood cells. They're deformed because the hemoglobin's deformed. Okay, so the protein inside of it's deformed, and it ends up a shape sort of like a crescent moon. We say sickle, like a sickle you use to cut, it's on the Russian flag, right? So this is what this is actually a microscopic picture of a red blood cell. See, it looks a little bit like a donut, but the hole doesn't go all the way through it. Okay, so what do you call it? Thumbprint cookie or something like that has that kind of impression, indentation in the middle right there. So, and they're nice and round, that's beautiful. Think of them kind of like, this is the way I like to think of them. You know those tubes that you go down the river in that have a bottom on them? Mm-hmm. So you sit in the bottom, you're going down the river in this tube, you're like a red blood cell, okay? You're the oxygen being carried through the river, yeah? So that's kind of what it's like. So think if you went through a tight tube kind of thing, your inner tube would kind of squeeze up around the tube and go through, right? So that is the way they're shaped, they're able to be squeezed around like that. If they're sickle shaped, they look more like this. Therefore, when they're going through the tube, known as a vein or an artery, they get caught and stuck in there. All right, and it causes that to be a pretty serious disease that takes place in some humans. Normal red blood cells, see they're going through the river thing, they're squeezing through here, their normal hemoglobin right here. Sickle cell, their hemoglobin's all stretched out, it's not the right shape, and they get caught in those small little vessels right there. Honestly, I'm not quite sure how they treat people. It's a genetic disorder. Oh, we have a video. Maybe that'll tell us how we can treat it. Here we go.
What shape are your cells? Squishy cylinders? Jagged zigzags? You probably don't think much about the bodies of these building blocks, but at the microscopic level, small changes can have huge consequences. And while some adaptations change these shapes for the better, others can spark a cascade of debilitating complications. This is the story of sickle cell disease. Sickle cell disease affects the red blood cells, which transport oxygen from the lungs to all the tissues in the body. To perform this vital task, red blood cells are filled with hemoglobin proteins to carry oxygen molecules. These proteins float independently inside the red blood cell's pliable donut-like shape, keeping the cells flexible enough to accommodate even the tiniest of blood vessels. But in sickle cell disease, a single genetic mutation alters the structure of hemoglobin. After releasing oxygen to tissues, these mutated proteins lock together into rigid rows. rods of hemoglobin cause the cell to deform into a long, pointed sickle. These red blood cells are harder and stickier and no longer flow smoothly through the blood vessels. Sickled cells snag and pile up, sometimes blocking the vessel completely. This keeps oxygen from reaching a variety of cells, causing the wide range of symptoms experienced by people with sickle cell disease. Starting when they're less than a year old, patients suffer from repeated episodes of stabbing pain in oxygen-starved tissues. The location of the clogged vessel determines the specific symptoms experienced. A blockage in the spleen, part of the immune system, puts patients at risk for dangerous infections. A pileup in the lungs can produce fevers and difficulty breathing. A clog near the eye can cause vision problems and retinal detachment. And if the obstructed vessels supply the brain, the patient could even suffer a stroke. Worse still, sickled red blood cells also don't survive very long, just 10 or 20 days, versus a healthy cell's 4 months. This short lifespan means that patients live with a constantly depleted supply of red blood cells, a condition called sickle cell anemia. Perhaps what's most surprising about this malignant mutation is that it originally evolved as a beneficial adaptation. Researchers have been able to trace the origins of the sickle cell mutation to regions historically ravaged by a tropical disease called malaria. Spread by a parasite found in local mosquitoes, malaria uses red blood cells as incubators to spread quickly and lethally through the bloodstream. However, the same structural changes that turn red blood cells into roadblocks also make them more resistant to malaria. And if a child inherits a copy of the mutation from only one parent, there will be just enough abnormal hemoglobin to make life difficult for the malaria parasite, while most of their red blood cells retain their normal shape and function. In regions rife with this parasite, sickle cell mutation offered a serious evolutionary advantage. But as the adaptation flourished, it became clear that inheriting the mutation from both parents resulted in sickle cell anemia. Today, most people with sickle cell disease can trace their ancestry to a country where malaria is endemic. And this mutation still plays a key role in Africa, where more than 90% of malaria infections occur worldwide. Fortunately, as this adaptation thrives, our treatment for sickle cell continues to improve. For years, hydroxyurea was the only medication available to reduce the amount of sickling, blunting symptoms, and increasing life expectancy. Bone marrow transplantations offer a curative measure, but these procedures are complicated and often inaccessible. But promising new medications are intervening in novel ways, like keeping oxygen bonded to hemoglobin to prevent sickling, or reducing the stickiness of sickled cells. And the ability to edit DNA has raised the possibility of enabling stem cells to produce normal hemoglobin. As these tools become available in the areas most affected by malaria and sickle cell disease, we can improve the quality of life for more patients with this adverse adaptation.
All right, well, there you go. That answered all the questions, didn't it? Yeah. Okay, so this is what the normal hemoglobin looks like. It has 4 tertiary folded proteins, and each one of those can hold what is known as a heme. All right, this little slab looking thing right there is a heme, and on the heme is an iron atom. And the iron atom holds on to an oxygen. So that's how that works. Think about when we talk about iron rusting, right? Because oxygen will attach to it. So that's what's happening here. And so the oxygen attaches to that, and it takes it to where, of course, we need it in each one of our cells. When it becomes depleted, of course, it needs to go back and get some more oxygen, and therefore the red cell goes back to the lungs to be re-oxygenated. All right, the white blood cells have a little more intricacy than the red blood cells. Leukocytes exhibit amoeboid movement. All right, they can move. They can squeeze into tissues and squeeze around things out of a blood vessel. That's called diapedesis. And that's fortunate because what they're going to do is they're going to squeeze out of the blood cell and go to where some bacteria are, and they're going to gobble them up. Depending on the type of white cell, we're going to see how they operate. Chemotaxis is what that is. Okay, so attraction to cells, I should say maybe, there, it does say attraction of cells to chemical stimuli So they go to the rescue. So it's chemicals from a foreign object that that encourage them to squeeze out and go attack, okay? Because they can recognize something that's foreign, something that's not made by your own body. When we talk about the next module, when we get into the immune system, we'll see sometimes when your cells and your white cells attack your own body thinking it's foreign, that's called this autoimmune disease, right? So those are when we attack ourselves. All right, so chemotaxis is just think of that like a taxi. It's going to take it, attract it to a chemical, and move toward it. Phagocytosis, you've probably heard of this. It means cell eating. A lot of the white blood cells function by engulfing and digesting these foreign cells, bacteria, things like this. Interesting, sometimes viruses can kind of circumvent this procedure and sneak in, and the white cells are unaware that they're there. That's why we can treat bacteria with antibiotics, and we can't treat viruses because they're not alive, right? They're sneaking in there. There are several specialized types of white blood cell. Some are called granulocytes. That's because when we stain them, we see large granules, and there are actual chemicals inside those granules. Others are called agranulocytes, meaning they have no visible granules when you stain them. All right, and I'm hoping to get a sample of your blood to stain, and we'll be able to see. I had one time a student that had a really awesome granulocyte in there called an erythrocyte, and it was beautiful under the microscope, beautiful. They're normal to have, but I'll talk to you about them in detail here in just a second. All right, do you have something somewhere that, does it tell you to list the types of white blood cells? Is there like a chart or something?
Yeah. 5 types.
The 5 types, is that next?
Yes.
Okay, cool. Because here they are. All right? So these are all the rest. So hold on just a second. I guess the next slide will be what you want. We have red blood cells, erythrocytes. We have platelets, thrombocytes. And these are the 1, 2, 3, 4, 5 different types of white blood cells. Okay, these they're blue like this because they've been stained with a blue stain. All right, so I want to take you to the next slide. You're going to see, I like these better, this picture better. I like the sizes they show. And right here, these 3 on this side are the granulocytes, meaning that when you stain them, you can see some granules in them. And we're going to see there's a neutrophil, the basophil, the eosinophil. Those are the granulocytes. Then we have the big one, the monocyte. If you've ever known anybody who had mononucleosis, mono, the kissing disease. I knew a little kid, she was 6 years old, she had mono. I don't think she was kissing anybody except for mom and dad. So it doesn't necessarily mean that's the only way you get it. It is a way of passing it, however. Anyway, they're massive. They're very, very large. And then the smallest one is actually the lymphocyte, which has a quite large nucleus, you'll notice there, and a smaller cytoplasm. The lymphocyte is very, very specialized. We'll spend a lot of time talking about the lymphocyte in the next module when we talk about immunity. Lymphocyte has a complex immune response versus these other guys, which pretty much gobble up bacteria. Okay, so the most common, so what I had to do at the vet clinic was I would take a blood smear and do this thing called a differential, a diff slide. I'll show you how to make one. And you end up counting, like I had a little counter and I'd look in the microscope and I'd count 100 cells, 100 white cells. And I would have, are they neutrophils, are they basophils, are they eosinophils, are they monocytes, are they lymphocytes, and I would count. By far the most common one is the segmented, it's called a segmented neutrophil, segmented because its nucleus is quite segmented. And the second most common would be the lymphocyte. Both of those very easy to see. Monocyte's massive, so I wouldn't see those very commonly. Basophil, see that kind of looks like a letter B in there? We call those a band. It was another word we used for those. And then we have the eosin. The eosin's the one that shows the granules. These are called granulocytes, but in my personal experience, I didn't see granules in those. I only saw granules in the eosin. And they're beautiful, beautiful. So let's talk about our segmented neutrophils. You can see one right here. It's all segmented there. Sometimes it looks like a pair of headphones that are in there that you'll see. They're the most common. They are responsible for phagocytosis. They engulf up to about 10 bacteria, which it's not that many bacteria, because bacteria can multiply very quickly. And that will, then they'll die. And that's what pus is. So when you see pus in a wound, it's all a bunch of dead white cells. All right? Sometimes it's sort of yellowish looking because they gobbled up a whole bunch of bacteria in there. These, by the way, are red blood cells next to them. See how much bigger the white blood cell is, the red blood cell? And notice it looks like it's got a hole in it, but it doesn't really. It's just really thin in there. Okay, so that's our neutrophil. Our basophil, another granulocyte. They're fairly rare, pretty large. The little granules in there are called histamine. Histamine is the stuff that causes inflammation, namely in your eyes and in your, like my nasal passages today, making me want to sneeze. It's a chemical that does that. Now, why would we want this chemical that makes my eyes itchy and my nose run? Well, it's because it's bringing white cells. It's bringing white cells over here. The inflammation will cause more blood to come there. More blood, more white cells. I can take care of whatever it is. Typically for us, the thing that's happening is an allergy. I'm allergic to something. And so really the white cells don't do much because my allergy is not because of a bacteria, and my allergy is because of pollen or something like that. And so they'll release the histamine. So if we are really being bothered by it, what do we take? We take a antihistamine, right? And it causes that histamine not to go binding to our tissues, making our eyes red and our nose running and that kind of thing. In my experience, and histamine also makes me very, very sleepy. So if I take it, I'm just like, meh. So I'm numb. I can take it at night when I'm sleeping. It also has something called heparin in it. Heparin is a chemical that keeps your blood from clotting. If you've ever had somebody take a blood sample from you and they put it in a little purple top tube and they go like this, the purple top tube has heparin in it, and that keeps it from clotting, because sometimes we need to do tests on the red blood cells of your body, right? We need to test those. So we'll keep it from clotting so we can test for that. Also, if you've ever had an IV in your arm, sometimes you'll hear the nurse refer to it as a hep lock. It's a heparin lock. It's got heparin in there to keep it from clotting right in there so they can come back and put some injections in later. Or put fluids in there.
Blood thinner is, or is that?
It's a blood thinner, absolutely. It keeps it from clotting. So it speeds up healing by bringing lots of oxygen, bringing a lot of white cells, immunity factors, including what lymphocytes bring. The eosins show very large granules, super cool looking. They actually help decrease inflammation, and these typically show up if you have a parasitic infection. So if you go to another country on a missions trip and you get a creature that crawls into your eye and you get Chagas disease or something like this, you're going to show it a couple months later by a doctor doing a test on you and saying, man, you got a lot of eosins in there. Where have you been? Where have you been traveling? Humans don't get that many. Okay, I'm going to take that back. Americans don't get that many parasitic infections around here. But you know who does around here? Dogs. So I saw them all the time because of dogs. There's a certain kind of a disease called Ehrlichia, and dogs would get it. So a dog would come in, and the symptom was they had a nosebleed. And the bleeding wouldn't stop. So I'd get the blood sample to go test it, blood from their vein. I'd test it, and I'd see tons and tons of eosins. Then we could treat it based on symptoms and blood test that the dog has Ehrlichia, and we use certain medications for treatment of that. All right, the lymphocytes are probably the coolest of all. Notice their small, large nucleus right here. And the reason is that they are not only made in the bone, which they are, all cells are made in the bone, all blood cells, including all white blood cells, are made in the red bone marrow. But the lymphocytes actually can proliferate, grow more of them, by taking the cell and going to a place called a lymph node. That's what lymph nodes are. The lymph nodes are just little spikes. There's lots of them here in your face because guess what? I'm being exposed to a lot of stuff up here. I've got them in my armpits and all of my joint areas. And those are just patrolling. Blood goes through them. And as the blood goes through them, it checks the blood. And it says, anything wrong with the blood? Anything wrong?
Like,
and if there is, it can make certain chemicals.
like you got a bee sting right here, and then this one swells up real big because the blood went through it and it caught the bad stuff.
Yeah, yeah, absolutely. And so, like, even if you get a cold, and so, like, I noticed with my son one time that I could see, I was looking at him from behind, and I could see bump, bump, bump. I said, oh my goodness, he has all these bumps in his neck. I took him to an ENT, an ear, nose, and throat doctor, and he said, oh yeah, it's all of his lymph nodes. Is he sick? And I said, well, not really. And so he did a blood test. He goes, yeah, he's sick. He's got a little cold. And he goes, that's why that's there. He just had a thin neck, I guess. And you could see him. But we're going to see in the next module how the lymphocytes will go through a super complex process to make specialized disease fighters called antibodies. Antibodies.
So lymph nodes really important.
Oh, so important.
Because
Yeah.
I knew twins who were born, and one of them like had a really small amount of lymph nodes,
Interesting. The actual nodes themselves, huh?
Yeah, so they couldn't be around people and they had a lot of doctor visits to do that.
So they had a poor immune system.
Yeah.
Yeah. So, yeah, our immune. Yeah, it's interesting. I've never heard of somebody just being born with fewer lymph nodes. How about that? Okay, so what we can see here, right here, we're going to see how antigens will cause the lymph node to proliferate, making more lymphocytes. The lymphocytes will go out. We have B cells, we have T cells, killer cells, all this stuff. That all are lymphocytes, and that's the lymphatic system, which we'll cover in another module. The monocytes, huge. Look how big they are. All right, and we get a proliferation of those with certain viral infections, namely the one called mononucleus, just a virus that attacks. All these monos proliferate because of it. They can gobble up to where the segmented neutrophil can gobble 10 bacteria. These gobble 100. So these are the big eaters. All right, hemopoiesis, we know about that, the process by which formed elements of the blood are made in the body. That means the red cells, white cells, and platelets. It starts in the red marrow of long bones where hemocytoblasts, stem cells, differentiate into the type of blood cell needed by the body. So here we are in the bone. Okay, some of them are going to go this way. The special ones are going to become lymphocytes right here. Then we see the other cells. Most of them go this route to become red blood cells. Some of them fall to pieces to become the platelets. And then we see the other white cells right here. Here's our monocyte, has a kind of a special step in there, the big eaters. And then, of course, our very, very special lymphocyte over there, which can make these new chemicals. So the long bones are the most common places where those are found because that's where our red marrow is found. And so it's important, like there are people who don't have any arms and legs, right? So they have fewer long bones, right? So they're going to have trouble with blood manufacture. There is still red marrow in there to manufacture that. All right, good. So hemostasis. Okay, so this hemostasis is keeping everything even, right? Keeping everything even. In this particular case, it's going to stop blood loss. Okay, it's going to stop blood loss through the process of blood clotting. You don't want to bleed out. Our body is designed that we don't bleed out because we have this process of blood clotting. So of the module this week, this is the most complex portion of the module, okay? So hemostasis. There are 3 stages of blood clotting. Number 1, vasoconstriction. What that means is the veins get smaller. Constrict means to get smaller. Vaso has to do with veins. It's a reflex contraction after trauma. Smooth muscle closes a breach. So we're going to see in a minute that the vessels are made of a tunic of smooth muscle. If there is a breach, in other words, a cut, or some sort of damage, even bruising kind of a thing, then what will happen is those little tiny vessels will all try to stop the bleeding by just closing up. So they can do it in small cases, right? So, and it says here that the jagged cut heals faster than like a knife, right? Jagged because they got all those little spaces where all that sharp edges, the platelets come rushing. If it's a nice clean edge, like some you do in a surgery, they're not as aware of it because there's less surface area right there. They'll still come and they'll still cut, they'll still clot.
So why does like when you like just bump somebody, you get a bruise pretty quick, and then if you like really like knock yourself, why does the bruise take longer to appear if it's like
If it's deeper, it's just lower down. You
so
can't.
it just takes it longer to
Takes that time, yeah. So that's called a hematoma, which is just a bruise is just bleeding beneath the skin. It's not coming out. All right, so it's bleeding. It's just underneath.
It's like all your capillaries got
Sometimes even worse. Sometimes it could be an internal bleed. If it was a big vessel, normally, like if a car accident, like a seatbelt kind of a situation, will grab hold of you and maybe break a vessel. Typically, when you have an internal bleed, it's an organ, namely the spleen, that starts to bleed, because of trauma. I've heard of football players have been hit so hard that their spleen starts to leak, and it's leaking blood. Yeah. Number 2 is called the platelet plug stage. All right, the platelet plug. The platelets swell up and get sticky. So the trauma, the cut, the whatever, exposed collagen fibers, those collagen fibers being open to the bloodstream that says that's not normal, that triggers the release of something called thromboxane at that cut level. And that thromboxane is an alarm to the platelets. Come on, come over, come over, come over. We need you here. So a thrombus will form. That's the clot. If a clot forms when this is not happening, we call that a rogue clot. It can form in a rough vessel. Sometimes that rough vessel will have some deposit on the inside of it for some reason, namely like a cholesterol situation, or there's something wrong with the inside. Instead of being nice and smooth, there is I keep using the word sharp, there's just edges there. And so then that will lead to the blockage of a vessel, causing heart attack or stroke. One of these clots can break loose, traveling to somewhere else, causing something known as an embolus that can go to your lungs and drop you like that. Boom, you're dead. Just like that. So it's good to keep your blood from clotting unless it needs to be clotted. Number 3 is called coagulation. This is a cascade of biochemical events. Oh, when I say that, cascade of biochemical events, that's when you say, no. That's the physiology portion here. We'll talk a little bit about it. And you can see how it gets you will see how it gets very, very complex. All right. This is an embolus right here. We have, see the roughness on the inside of my vessel right here. Then we have platelets kind of cover it up. They're thinking it's some sort of cut or something. They cover it up. And then some of those platelets will break off as a clot. So that's just like some blood cells that have been surrounded by platelets, if you will. And if they go up into here and they get caught in the lung right there, they'll cut off the blood supply to part of the lung. Not everybody drops dead from that, but depending on how big the clot is. Sometimes it'll just be it's called a pulmonary embolus. It's not good. Okay. Thrombus is the platelet plug designed to stop bleeding. An embolus is a blood clot that is loose in the bloodstream. Not supposed to be there. There's a test called the D-dimer test, which can give you an idea if you have clots floating around in your body. And if you do, they want to find out where in the world are they.
How does it tell them if you have clots?
You know, I'm not sure what trips it. I'm not sure what it is. I just know that they can get a number from it and they say, oops, we think there might be some clots in your body. Now we're going to go do like a scan of your leg. A lot of times it'll be in the legs. You'll have like somebody gets like a swollen leg, and it's because they have a little clot down in there, which can resolve itself. Notice how this is going through the heart to the lung. It's not going on the heart. The heart has blood vessels on it. When the clot's in that causes a heart attack. The heart attack doesn't come from a clot going through the heart. It's on the vessel feeding the heart that causes a myocardial infarction. Okay, so I just want you to take a look at it here for a second. This is the cascade of biochemicals that takes place in clotting. All right, so all this has to happen. What we're trying to get to is X, which is really 10. Okay, X is the Roman numeral 10. I call it X because it's easier to do it that way. So what happens is we have some sort of trauma, all right here. And so what happens is I have a chemical that causes blood clotting agent, is that 7? 7 to turn into 7A. And 7A then in turn gets turned into 8A. So is that 8? To 8 to 8A. Then we get down here, we finally get to 10. And if I have enough chemical, because the condition is pronounced enough, then I'd call X or 10 to go to 10A. And then I have 5A in conjunction with 10A, which will cause prothrombin to convert into thrombin. And thrombin can then, is at 2A, makes fibrinogen 1 turn into fibrin 1A, which influenced by 13 turns into 13A, I get a clot. Whew. It's very complex. God's showing off once again, but he's doing it for a reason here. Okay, so here, this kind of simplifies it a little bit right here. I've got my trauma, because I had the other route too. I have an intrinsic pathway, damaged surface, I have trauma here. Depending on how I get traumatized, I'm going to go through all these steps. So my steps, we have something called von Willebrand factor that makes 8 and 9 turn into 10, influencing 5 to make thrombin turn into fibrin, and fibrin makes the clot. Okay, there have been people that have studied this in detail and have become believers in God because of it. They said there's no way that all of this, if every step here took millions of years to develop, we're all dead. We're all bleeding out. We don't have enough clotting factor. This had to be created at once for it to work properly. Okay? So therefore you can see the fingerprints of a creative God here. All right, this is what the clot ends up looking like. Okay, so the fibrin that's made, it literally makes this net that holds on to the red blood cells and keeps them in place. You've all seen a blood clot before, right? It's like a little collagenous thing sitting there, and it's because the fibrin's holding on to it like a little net. And that gives the time for the tissue that's been damaged to heal. And once it's healed, the clot gets taken apart and can be disseminated that way. Let's watch a little video.
The process of hemostasis has many components and interacting mechanisms that often appear complicated and difficult to grasp. Multiple factors activate each other from different pathways under the control of various enzymatic reactions in a very convoluted manner. However, if looked at from the perspective of what happens at the injury site to specifically stop bleeding, it is in fact quite simple to understand.
Quite simple.
Only two blood components emerge as necessary, platelets and fibrin. The following animation will highlight the process by which they work together to achieve hemostasis. The endothelial layer in healthy individuals serves to protect against spontaneous accumulation of platelets and fibrin in the vessel wall. In the event of vessel wall injury, bleeding will occur. Hemostatic plug formation is initiated upon exposure of the subendothelium at the injury site. Vasoconstriction in the region of injury results from the release of thromboxane A2, a major enzymatic product of platelet activation which causes vessel wall constriction. Von Willebrand factor is secreted by the endothelial cells and binds to exposed collagen fibers at the wound surface. Platelets then adhere to the wound through their interaction with bound von Willebrand factor. This process constitutes platelet adhesion. Adherent platelets are activated and attract other platelets through a process known as platelet aggregation. Fibrinogen molecules form bridges between adjacent platelets. This forms an aggregate of platelets that occludes the wound and eventually stops bleeding. This unstable primary plug of loosely aggregated platelets must then be consolidated into a more stable plug. This is achieved through the generation of thrombin at the wound site. Free tissue factor released from subendothelial stores and platelets initiates the coagulation cascade with its interaction with activated factor 7. The complex of tissue factor and factor 7A converts factor 10 to its active form, factor 10A. Tissue factor 7A also converts factor 9 to its activated form, factor 9A. However, further generation of factor 10A is inhibited by the tissue factor pathway inhibitor. At this point, the amount of factor 10A produced is insufficient to sustain coagulation. Further factor 10A, to allow hemostasis to progress to completion, can now only be generated by the factor 9A pathway. By this stage, though, enough thrombin has been generated by the factor 10A to activate factors 8 and 5, which both act as potent catalysts. Factor 8A increases the capacity of factor 9A to activate factor 10 to 10A many thousand times. The increased factor 10A produced in this way, along with its own cofactor activated 5A, forms a complex that promotes the efficient conversion of prothrombin to thrombin. It is thrombin that then allows the conversion of fibrinogen to fibrin. While the process of the coagulation cascade as outlined can be confusing and difficult to retain, it is most important to remember the importance of the final result, fibrin, in wound healing. Fibrin is the biological glue that eventually seals the hemostatic plug and ensures hemostasis. As soon as a small amount of thrombin is formed, the clotting process accelerates and provides more and more thrombin into the wound. At high concentrations, this thrombin quickly converts fibrinogen to fibrin on the surface of the platelet aggregate to stabilize the hemostatic plug. Over the course of the subsequent 7 to 12 days, the process of fibrinolysis dissolves the fibrin in the wound as the site of injury heals, and the cell layer in the vessel wall is restored. Some scar formation occurs, and within a few weeks, the wound is completely healed.
All right, see that complexity? Yeah? So very complex.
You have
So,
a scar, that's from when we went over skin, right? And they just didn't come back all the way.
yeah, so she's talking about the scar tissue on the blood vessel itself. There'll be a little scar, but it typically gets, kind of like the bone, gets eaten away and reformed. Yeah, so it you don't necessarily, it's really, when we have scars, it's usually the skin that's scarred. Okay, so what do I want you to know about it, right? So I'm not gonna make you recreate that whole, cascade of biochemical events, all of the factors between 1 and 14 or whatever they were, and all the A's and all that kind of stuff. But what do I want you to know? Okay, so here's what I want you to know. We have trauma of some sort, and the trauma will take prothrombinase, which is already there, and make thrombin. And the thrombin will take fibrinogen, which is already there, and turn it into fibrin. All right? Oops, let me go back to that. Yeah, here we go. And that fibrin will make the clot. For all of these processes, that's a lot less, isn't it, of stuff. So it goes trauma, prothrombinase, thrombin, fibrinogen, fibrin. I'd like you to know those 1, 2, 3, 4 chemicals or proteins. And vitamin K is very useful in making this process go smoothly. And it's interesting, vitamin K is something your body is making. It's making it in your intestines. When we talk about the digestive system, we'll talk about this. Okay, so what happens is in the maybe it's the large intestine. Yeah, the large intestine, there are reactions taking place for vitamin K to be synthesized. There are 3 types of vitamin K, K1, K2, and K3. And the one we need for helping with the clotting is made in our bodies. You may take a vitamin K supplement, it's probably one of the other vitamin K, K2 or something. So it takes about 8 days after a baby is born in order to produce vitamin K. All right, so prior to 8 days, a baby doesn't clot very well. And this is why the Bible tells the boys not to be circumcised until the 8th day, because the vitamin K hasn't been, so God knew that. How about that? Pretty cool. I remember we had a dog one time come in. It was a big dog, and the people complained. They said, well, he's got blood, his nose is bleeding. So first of all, we thought, well, maybe he's got this Ehrlichia thing, and so we weren't sure. He was acting strangely, this kind of thing. His nose wasn't bleeding all the time. So I was told to go get a blood sample. I went and got a blood sample from the dog. So typically you go to their arm, you get the blood sample out, then you put just a little tape over that. I'm testing at first I'm just doing regular tests on the blood. And then the doctor calls me back. She goes, take a look at the dog. We're all standing there looking at the dog who's standing in his kennel. He's happy as can be. And he's just bleeding out of where I took the blood sample. And so we're all standing there, and I said, man, that dog doesn't have any clotting factors. And so the doctor goes, oh, good point. Let's test those. So I tested it. So, I told you about the clot tube, and you spin it down, and, well, it didn't clot. So it turned out this dog had something that was keeping its blood from clotting. And right off the bat, we asked the owners, have you put out any rat poison in lately? They said, oh, yeah, we sure did. The dog had eaten the rat poison.
Oh.
Yeah. We were able to treat him. Yeah, we were able to treat him and all that stuff, and we were able to save the dog. Yeah, the way that the rat poisoning, that's how the rat poisoning kills the rat, is that it has the poison that they won't be able to stop their bleeding, and they also have diatomaceous clay in there, which cuts open their stomach. So their stomach cuts open and they bleed out. Yeah, it's kind of sad. It's very effective. All right, so we're looking here at a blood flow, right, just flowing through there nice and smoothly through a vessel. As long as everything's okay, it'll just flow through just like we're in a nice stream with our little tube going down there. What happens if the blood flow slows down, though? If it doesn't keep going, if somehow it's sluggish, we will go into coagulation. All right, so it's important for the blood flow to be good. What that means is the amount of water. Don't get too overly dehydrated. People can start getting clotting if they're not hydrated completely. Okay, blood types. You probably know the blood types. Does anybody here know what your blood type is? Do you know? Nobody knows? You know?
I believe I am O positive.
Oh, me too. It's so boring.
I think I'm B.
That's awesome. I hope somebody here is something besides O, because O doesn't show it. That's just O's fine. O's the most common, by the way, Mateo. We're the most common out there.
I think the rarest is AB.
I'd have to look it up. I'm not exactly sure. But there are 4 types: A, B, and O. By the way, AB is AB because neither A nor B show any dominance over each other. But both of them show dominance over O. O is super recessive, which is interesting that that would be the most common type, the recessive one. About 40% of the population is type O.
What's the difference between the types? Like, what is?
You're going to see right here. It's determined genetically based on what your parents have. They're differentiated by this, surface antigens. All right, so why do they have the surface antigens they have? We do not know. We don't know why we have that. All right, but they have, so in other words, if you have type B and I have type O, my surface antigens are different than type B's surface antigens. Okay? The outside of it looks different. The surface antigen will trigger production of an antibody. And antibodies are made in the specialized white blood cell called the lymphocyte. All right, another surface antigen is called Rh factor. And by far, most of us have that antigen on the surface. So in other words, I am O positive. Positive means I have an Rh factor on the outside of my cells. If you're Rh negative, you're completely naked on the outside of your cells in reference to blood type. All right, so here we go. If somebody is type A, they have their blood cells like this with a little surface antigen that looks kind of like, I mean, this is just a fanciful way of showing it right here, that is anti-B, right? So if you have type A and somebody tries to give you type B blood, you already have antibodies floating around your body that will attack it and cause it to coagulate. And then you're going to have a bunch of clots all over your body and you're going to die. If you're type B, then you have these surface antigens and antibodies against A. If you are AB, you have both surface antigens and no antibodies. So type AB can actually receive blood from anybody. Type O has no surface antigens but both types of antibodies. So if you try to put this blood into a type O person, all these antibodies will cause that blood that just came in to be coagulated. If I take O and put it into these guys, it doesn't matter at all, it's naked. So O is the universal donor. Yeah.
Is it any O, like positive or negative, or is it?
If you are O positive, then you can give to anybody who's Rh factor positive. If you are O negative, you can give anybody. If you are O negative, you can only get O negative blood. You can't get O positive blood. We'll talk more about that in the next module when we talk about the way that all these antibodies, how they work. Okay. So here's just a Punnett square to show if your dad is type A, he might have an O in there because A is dominant to O, B is dominant to O. Here's if mother's type B, but she has an O gene, and dad's type A but has an O gene, you can have 25% of the kids will be AB, 50% will be, 25 will be B, 25 will be A, and 25% will be type O. And that's because A and B it's incomplete dominance. And when it comes to the Rh factor, since the vast majority of the population is Rh positive, I'm going to say like 95% of the population is Rh factor positive. The only time it comes into play is if you have an Rh negative mom and the baby is Rh positive, assuming dad is passing an Rh positive gene to the child because it's that dominant, then we have a problem here that the woman actually starts making antibodies against her own baby, and that is serious because she'll kill her own baby, all right, with her white cells. And so the way they deal with this is that they test the mom. Women all should know if they're Rh negative. I had at least one student who did the Rh factor in my class to find out she was Rh negative. So she didn't know before that, but she was. Okay, it really only happens in the second pregnancy. The first pregnancy is not a problem because there's no antibodies made against it yet. You have to encounter it before the antibody can be made. Okay, that's sort of like the same function of a vaccine. A vaccine, you encounter it, you make an antibody, so when it comes to you again, you'll make the antibody against whatever that disease is. Okay. All right, we're in part B now. We're going to talk about the heart. Circulatory system, once again, heart to lungs, back to heart to body. All right, humans have what is known as a closed circulatory system. Arteries carry blood away. Arteries away. You might be tempted to say that arteries have oxygen-rich blood, and in most cases they do, but not in every case. Oh, that's awful. Arteries away. Oh, it's all right. It's carrying blood away from the heart. The vein is to the heart. Right. Veins carry blood away. I'm sorry, arteries away, vein 2. Capillaries are thin, tiny vessels where gas exchange takes place. So essentially what we've got is in the heart we have large vessels, large arteries, large veins, and they travel down until they get smaller and smaller and smaller and smaller until they get to the smallest vessel called a capillary. If you look in your eyeball and you see little blood vessels, those are capillaries, very tiny, so tiny that only one red blood cell at a time fits through, which is very efficient because as it fits through, it's going to be squeezing through, and as it squeezes through, it's touching the side and letting that oxygen out. It's pretty cool. So gas exchange, CO2 and O2. Pulmonary circulation from the heart goes to the lungs via something called the pulmonary artery, from the lungs to the heart via the pulmonary vein. So if it's going to the lungs, it's going there for a reason. It's going to get oxygenated. Which brings up an interesting thing. When we talk about deoxygenated blood, we always color it blue. You need to understand that blood isn't actually really blue. Our veins are blue, and you can see them a little bit, and they look a little blue. So people for a while thought the blood was blue. I will say deoxygenated blood is dark red, like a purplish red. And if you cut yourself, you know how bright blood is, right? Guess what it's touching? Oxygen. The oxygen is just hooking up to it, boom, just like that. That's why we call that frank red. Systemic circulation from the heart to the body via the aorta, and from the body to the heart via the vena cava. So the arteries, the aorta is taking it away. The aorta is the largest of the arteries, and the vena cava is the largest of the veins. So the closer you are to the heart, in the systemic circulation, the larger the vessels will be. But there are vessels, of course, near there that need to be fed as well, including the vessels on the heart itself. All right, so when I show the blue blood, that is just showing us that it is deoxygenated, needs to get more oxygen into it. When I show the red blood, it's already been oxygenated. The reason why, did you ever hear that royal people were known as blue bloods? One of the reasons they called them that is because they were very, very intent on keeping their skin very fair. They wouldn't get in the sun and get a tan or anything, so they were very, very white skin, and you could see those blue veins really well. In fact, there were sometimes during Elizabeth's reign where they drew on blue vessels. So you could see these blue vessels, and they were known as blue bloods because of that. And now I don't think that we would find that attractive, right? So notice we have our figure 8 going through here, all right, because I want you to think of the heart like a double pump. All right, so here it is, made up of cardiac muscle tissue. Remember, it's the type of tissue it is muscular, and it's a very specific type of muscular tissue called cardiac. It's only found here, only in the heart. And I told you a long time ago, if you take a cell off of this and you took it by itself, it would be beating. It would be pulsating. That's how specialized it is. It's got a lot innervated. Weighs about 10 ounces for the average adult human. About the size of your fist. A little bit bigger. I don't have a very big fist. It's a double pump sending blood through the cardiac cycle. Heart rate about 75 beats per minute is pretty normal for a resting heart rate. We'll test that next week. I'll bring a stethoscope and we'll test our pulse and all that business. It's nourished itself, so keep in mind it's a muscle, and therefore it needs to have lots of blood supply. It needs to be very, very vascular. So the vessels that are actually on the surface of the heart right here are called coronary arteries and coronary veins. Covered by a membrane called the pericardium, and the large vessels include the aorta, leaving it, the largest aorta, like this, and the vena cava coming into it, very large, the largest veins. So you're going to be reading more about blood types, but I'm going to cover that in the next module with more detail, because that's when we're going to do the lab for it. I just got a message that my kidneys have been delivered. So I have kidneys at my house for y'all, eventually. All right, we good with this one? Have everything we need from this slide? All right, so the tissue itself, it looks a lot like skeletal tissue. In other words, it's got the striations in it, but it also has these little calculi in there. All right, these little things called the intercalcated disc. So you could, if you looked at a slide of smooth muscle versus skeletal muscles versus cardiac muscle, you should be able to tell the difference. If you're in medical school, you have to tell the difference. So it's in its own class. It's involuntary, striated, shorter cells than skeletal. They're joined together at these intercalcated discs. They're highly excitable. They can beat independently. So excitable meaning nerves touch them and make them beat. Okay, we're going to see specialized nerves inside. So if we look at it, here's some of those little intercalcated discs. You see them all along there. Okay, and then the stripes for sure. The internal anatomy, it says here, see page 340 in your textbook. Internal structure consists of 4 chambers. You must know this, gang. Our heart has 4 chambers, okay? So do all other mammals. Hearts have 4 chambers. 2 upper chambers, 2 lower chambers. They're divided by something in the middle called the septum. The upper chambers are called atria, right atrium, left atrium. Atria is plural. We have 2 atria. The lower, larger chambers are the right and left ventricles. We've heard the word ventricle before. In the brain, we know the ventricles were those little slits in there where the cerebrospinal fluid was made. The atria, that's the 2 of them, send blood to the ventricles. So if this helps you at all, it's like a baseball game. It goes from top to bottom. All right, at empty gravity, top to bottom. That's our cardiac cycle. They send blood out to the heart. So the atria will send it to the ventricles. So the smaller chamber at the top just sends it to the lower chamber. The ventricles send it either to the lungs, which is not very far away, or to the whole body, which is very far away. And then they're separated by these things called valves. We'll see the mitral valve, the tricuspid valve, mitral also known as bicuspid. Tricuspid means it has 3 little doors on it. And the semilunar valves look like moons. That's what lunar means, semilunar, half a moon. Really a crescent moon. Regulate blood flow to the aorta and the pulmonary trunk. The pulmonary trunk would be the vessels going to the lungs. Okay. So what I need y'all to do right now is take your fists and put them together like this. 2 fists. Okay, this is your heart. You're going to take your right hand and squeeze it, and then you're going to squeeze your left hand. Ready? Bump, 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 bump. So if you think about it for a second, when you contract your muscle, does it make any noise? No, there's no noise there, right? So when our heart is pumping, there's no noise there. But here's the deal, when it's pumping, it's going through a little door called a valve. And the valve opens and lets it in, then it goes and slaps shut. So when we listen to our heart and we hear thump, thump, that's the valve slapping shut. And they go just a little bit, the lower valves, boom, boom, upper valve, boom, boom. Little bit off from each other. That's why it's a thud, thud instead of a boom. All right, so it's a double pump. One side is only going to the lungs. It doesn't have to go very far. The other side is going to the whole body. All right, there it is pumping. So pump, 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 slap shut, and then slap shut. All right, here's what the inside looks like. We have the blue side, which would be the deoxygenated blood, versus the red side, which would be the oxygenated blood. So where is the blue blood going to? Remember, when I say blue blood, I'm not literal, right? Where does the deoxygenated blue stuff go? Where's it going? Where? In the lungs, right? That's the only place. So it's going from top to bottom, out through the pulmonary trunk, to the lungs, getting oxygenated, coming back in to the top. So remember, this is the pulmonary vein. Look, it's red. So even though but it's coming toward the heart. So this is the only case in which you're going to find a vein that is fully oxygenated. Most of the time, the veins are deoxygenated, unless they're right there, right? We're going to see it going out the aorta through the artery, the big artery going away with a bunch of oxygenated blood. Question?
So do the blood go through the pulmonary artery?
Yes, it goes, from the pulmonary artery, it's going away, right? Artery away. And so that is going to the lungs. So artery away, but it's deoxygenated because it's going only to the lungs to get oxygen.
Because
And then,
Ollie, when he was born, he didn't have his pulmonary arteries. They had to create them.
seriously? I'm going to talk to your mom about that. He didn't have any?
I think he's missing one.
Because it does split into pieces
Yeah,
right there.
and then so they had to make one out of certain.
So he had to have open heart surgery when he was a baby? I've never
He
known
had
that.
to go all the way to California.
Well, he obviously was able to live until surgery time.
Yeah.
Obviously, he's here with us.
Yeah.
Thank you, Lord. Yeah. Okay, so then it's going to come to the veins back into the upper chamber, which is the left atrium. Remember, it's according to me. All right, you're looking at, this is left, this is right, because it's not according to how you're looking, it's according to me. And then the left ventricle will send it through the aorta to the whole body. So the left ventricle has to have a stronger muscle because it has to pulse it all the way down to your toes. All right, so when we open up our heart next week, you're going to be able to tell the difference between the right ventricle and the left ventricle by the size of the muscle. The muscle will be thicker on the left side. Here you can see some of the valves. Here's our tricuspid, see the three? And our mitral, also known as the bicuspid, like that. Then we have, so these are the upper valves right here going to the upper chambers or coming from the upper chambers. We can see right here. So when we go thump, 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 thump. So slap close, slap close. That's the thump, thump. Oh, this shows it a little bit. Ready? So pump, pump. See, they're close to each other, but not quite. And that's because they're in order from going to the top to the bottom. All right, so when you look at a normal valve, this is a normal tricuspid valve right here. When it's closed, it's closed until blood is flowing through it. When it opens like this. Close, open, close, right? If there is something wrong with the valve, if these parts don't fit together very well, sometimes when it's closed, blood can still get through that. And if that's the case, if you have a problem known as a heart murmur, that's because one of the valves isn't quite closing all the way.
My brother had that when he was born.
Does he not still have it? So he just grew out of it? Okay, he just grew out of it.
Grow out of it.
Apparently.
Well, Ollie has to redo another surgery, and it's to fix it again.
Well,
Because they put a patch over it, and it was his baby.
We're talking about a valve now or back to the pulmonary?
He has a murmur too.
Oh, he does have a murmur. Okay. Sometimes the murmurs are, you know,
I don't know where.
can cause deficits. Sometimes the murmurs don't cause any trouble at all.
Yeah, my mom has one, but it doesn't.
Yeah, it doesn't cause any problem.
Yeah,
Right.
when my brother was born, they said that he had it,
Uh-huh.
and for all of his checkups, and until he was about 2, my mom said like the only thing he would eat was avocados, and she took him for his checkup at 2. You know, they're every 6 months or whatever, and they said that it was gone, and then after it was gone, he didn't like it anymore. He doesn't.
I was about to say hooray for avocados, man.
Maybe his taste buds just changed. How does that have an effect on avocados?
I have no idea. I'm just saying, hey, we'll just use that
They
data.
said something about the fats or something. I don't know.
That's true. A lot of fat in it. Yeah. So, so if you're listening to somebody and you're listening to the heart and it goes dum, 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 that's good. But if you listen, it goes like this. That's a murmur.
That's how
Sounds like
it
a washing machine.
Yes.
Like that. So when I was examining dogs, I would come in on it. That's one of the puppies. I'd go in there and examine them. And if I heard I'd say, I'll have a doctor with you in just a minute. You know, so they'd come and listen to us. Sure enough, it's a murmur. And typically,
How do they fix that?
we don't have to do anything about it. Okay, typically, it's okay. If it's just a murmur. One of my sons went in for a physical because he played football with Troy. And he goes in, and the doctor tells him, well, you know, he has a heart murmur. I said, what? He goes, yeah, it's very slight. It's very slight. It's not going to be any problem. It won't affect his football at all. I said, okay, because he was like really fast, and he, was a good football player. But I wonder if later it might become a problem. So if it gets really bad, so here we see what's called as valve stenosis. Stenosis means stiffened. So it's not nice and pliable, right? It's all stiff. And if that happens where there's so much damage to that because, think about it. There's a big muscle pushing blood through it with a lot of force. And so over time it may damage it to the point where it's just simply, maybe like this the whole time and blood's just leaking through all the time. You don't have very good circulation that way. So there is something called a valve replacement. I have a friend who had this done, and he had to go I think he had the valve replaced, and he had to do the other side or something. He has some sort of valve disease or something in his body that's attacking the valves of his heart. And so you can see right here, this is pretty cool, because what they'll do is they'll bring up through the giant leg vein right here into the heart, and they'll go through the septum somehow, and they'll be able to put that artificial valve in here. And I have a little video that shows that. I can still do it like that. No sound. So you see it's coming up through there. This is coming from the other side. It's going to twist this, which opens, takes this off, and this little flower thing comes out here. It's going to open up. Then they're going to remove that blue thing. They're going to put stitches. You're going to see here. They're going to arrange it just right. Then they'll put some stitches. And they'll pull the blue thing out, which opens it up right down there at the bottom. And you'll see now here, they'll take that out, and that will be attached there with the sutures right there. And somehow it's able, I think it's under pressure alone, I think. It snaps shut normally, and then when the pressure hits it, it opens.
My grandma had that done.
Sure enough? Yeah.
My grandpa had that done, and then like 2 days later, he reacted really badly to it. He died.
Sorry. So he reacted badly to the valve?
Something about it.
Because sometimes even if you just have a regular surgery, anytime you get cut open for anything, you have an increased chance for blood clots, which could cause a heart attack. Okay.
So is that replacing a valve or the opening to the valve?
It's replacing the actual valve.
Is there, are you born with 3 or 2?
4. We have the 2 big ones and the ventricle to and then upper ones for the atria to the upper vessels.
He was born
Cindy Luna.
with missing one,
Wow.
and he has to have a service dog because he had a plastic one in there. They put or something, and it's not a catheter. It's something else, and he has to have a service dog because the dog can hear it when it starts going too fast, and it'll alert him, and he has to go to the. I don't know what it is, but he has to do it.
So his heartbeat goes too fast?
He says that when he's under, like if he's
Tachycardia?
swimming, you can hear it.
It's probably just that little plastic thing slapped,
Yeah.
because the blood goes through and then it's almost like a spring on a door. It'll snap shut.
Yeah.
And keep in mind, every bit of that artificial stuff in there, your body's going, artificial stuff, foreign, maybe making blood cells against it, white blood cells against it. That's called rejection. All right, the cardiac cycle. The blood is pumped through the heart via electrical commands. Both ventricles and both atria contract simultaneous with each other, but the atria contract just before the ventricles. And that's why we have thud-thud. The sinoatrial SA node sends signals to the atria, and the atrioventricular node, the AV node, sends signals to the ventricles. So A for our atria, V for ventricles. Step 1, we have something called early diastole. Funny word, diastole. Step 2, we have systole, the atria contract. 3, we have ventricular contraction, that's the lower chamber. And then ventricular ejection, sending that blood out to the rest of the body. And then relaxation, the atria begin filling again. So the atria fill up, get pumped to the ventricles, get pumped to wherever they're going. Crossover, one to the lungs, one to the body. That's known as the cardiac cycle. And we have these nodes. The SA node and the AV node is right here. All right, so, is this where I usually tell that story? Yeah, so if my, let's go back here. I had a friend who, when their son was born, he was full size, 8-pound baby, but he had over 300 heart rate. And he had a condition known as Wolff-Parkinson-White syndrome. And so what that meant was he had actually an extra bundle of nervous tissue. One of these, he had another node. He had a 3rd node in the middle of his heart right there, and it was causing it to short circuit. So if you listen to his heart, it went brrrr, like you can. They said over 300 because they couldn't even count it. All right, because it was too hard to count per minute, that kind of thing. And so what happened was that he would get, so if you have a short circuit, what we're going to have to do here is going to have electrical impulse, contract, electrical impulse, contract. What would happen is instead of doing that, it would just go back and forth between the two and cause the heart just to go like that, called fibrillation. And so when that was happening, of course, he's not getting good blood flow. And so that's a problem. And so when he was a baby, what they had to do with him is he had to wear a monitor all the time, and then the monitor would beep when it went up high. And you know, I'm kidding you not. This happened to a friend of mine. I saw it happen. They would turn the little baby upside down and put his head in a bucket of ice water. Like that, and it would shock him and cause that short to stop and to go back to the AVSA. Isn't that crazy?
Wouldn't it do the opposite?
Nope, it shocked it right out of there. And then when they told them that, they were like, "You've got to be kidding me!" And it worked every time. As he got older, he knew it was happening. He didn't have to have the thing. He said, "Oh, he called it episode." And the way he would fix it, instead of dunking his head, he would go stand on his head, and just the pressure coming in, the blood coming down to his head would knock it back into rhythm.
Do a handstand?
Just like bend over or
Headstand. He had to get on his head and put his feet in here.
Yeah.
So a headstand. Yeah. And so he later went to college on a football scholarship. Because what happened later, what they did was they came in and did something called like these radio waves that come in, and they took they came in through here and touched this with a radio wave and just obliterated that extra bundle of nerve tissues. Okay, let's watch a little video.
Veins are blood vessels containing blood flowing to the heart, while arteries have blood flowing from the heart. The blue is representative of blood vessels containing deoxygenated blood, while the red blood vessels have oxygenated blood fresh from the lungs. Now, let's follow the path of the blood through the heart. The superior vena cava receives blood from the head, neck, upper limbs, and chest. Meanwhile, the inferior vena cava receives blood from the trunk, viscera, and lower limbs. Both the superior and inferior vena cava end up in the right atrium, one of the 4 chambers of the heart. The heart not only has 4 chambers, it also has 4 valves. The purpose of the valves is to keep blood moving in the right direction and not flow backwards. Blood exits the right atrium through the tricuspid valve, so called because it has 3 flaps, and enters the right ventricle. The blood exits the right ventricle through the pulmonary valve and enters the pulmonary artery. Again, it is an artery because blood is flowing away from the heart, but it is blue because it lacks oxygen. The pulmonary artery then splits into the left and right pulmonary arteries, which go to each respective lung. In the lungs, gas exchange occurs. The blood discards carbon dioxide and picks up oxygen. Now, blood comes back from the lungs through the pulmonary veins, entering the left atrium. Next, the blood is pumped into the left ventricle through the mitral or bicuspid valve. Finally, the oxygenated blood leaves the left ventricle through the aortic semilunar valve, entering the aortic arch. The aorta, which is the largest of all the arteries, distributes the oxygenated blood to the rest of the body. The aortic arch has 3 major branches, which supply the head and arms with blood. Then the aorta curls downward behind the heart, forming the descending aorta, which descends through the chest and continues down through the abdomen. In the abdomen, the descending aorta splits to supply the pelvis and legs with blood.
Okay, so the arteries are pulsing. All right, that's why if you do this, you can feel it really well. And that's why if you're going to feel your pulse down here, you have to push kind of harder because your artery's deeper, right? And the arteries are deeper because if you cut your artery, what's going to be happening? You know, save the liver, right? It's going to be pulsating out. The veins are much higher up in your body.
That was weird.
This is an old video we're not going to watch. Okay, so let's talk about those blood vessels for just a little bit right here. The veins and arteries are composed of 3 tunics. We have the tunica intima, tunica media, and the tunica adventitia. The tunics and veins are much thinner walled, allowing for more blood volume, whereas the arteries actually have a layer of muscle, a tunic of muscle, smooth muscle right here. It's there too in the vein, but much thinner. This picture doesn't show it very well. Okay, capillaries have no tunics, allowing for easy gas exchange. That's capillary. Easy gas exchange. And arterioles are the artery version of the capillary, and venules are the vein version of the. So those are just other words for capillaries. The venule, capillary, arterial. They're going in like that. And so here is where the gas exchange takes place, where red blood cells really can only fit one at a time through there. Okay, so single file. So it's very efficient that way. All right, here's what happens with the veins. The veins have something called a valve in them, and that's because they're just being pushed back to the heart by the blood that's before them. All right, so it's pumping through the arteries, but veins, it's just really the pressure behind it that's pushing it back to the heart. And they have these little valves. I remember being told that, like you get out of the shower and you dry off with always dry like this way, right? And I was like, why the heck does it matter? Well, it's because of these guys. So they're going toward the heart, so the valves are going that way. So if I rub a lot this way, I can maybe do damage to the valves. It's not going to be that much damage, but that's why when you dry upward, you're doing less damage to your skin, the veins that are in your skin. Okay, what about blood pressure? It's exerted on the heart by the vessels. The increased pressure can, over time, can cause damage to the vessels. That's called hypertension, the silent killer, they call it. It's determined by measuring the maximum pressure called the systolic, that's the contraction, or the systole, and the minimum pressure called the diastolic, the relaxation, or the diastole. Normal blood pressure should be about 90/60. We'll take your blood pressure next week. I have a little cuff that you put on your wrist, and most people say the accuracy is pretty lame, it's not very great, but it will tell you if it's way high, and you might need to get checked out. High blood pressure, anything over 150, over 95. So that's the systole, that's the diastole. And my understanding is the second number, the diastole, is the one we need to be really concerned about. How does it work? We put a cuff on your arm. Notice this part of your arm, if you're sitting like this part of my arm is right about the same place where my heart is. That's why we do it there. All right, so we can have it as close to the heart there as we can. We cut off the blood supply, then we open it back up. So have everybody had this done before? And the cuff gets really, really, really tight, and it gets uncomfortable, right? And you're like, oh, and then they let it go, right? And so there's no flow at all. The nurse will have a stethoscope right here, and she'll hear, that's the first number. She looks at the pressure on that one. And then, the second number. That's how it works. Do we have time? Yeah, this is the last thing we're doing today, so y'all just bear with me. It talks about the blood pressure.
If you lined up all the blood vessels in your body, they'd be 95,000 kilometers long. And every day, they carry the equivalent of over 7,500 liters of blood, though that's actually the same 4 or 5 liters recycled over and over, delivering oxygen and precious nutrients like glucose and amino acids to the body's tissues. All that blood exerts a force on the muscular walls of the blood vessels. That force is called blood pressure, and it rises and falls with the phases of the heartbeat. It's highest during systole, when the heart contracts to force blood through the arteries. This is your systolic blood pressure. When the heart is at rest between beats, blood pressure falls to its lowest value, the diastolic pressure. A typical healthy individual produces a systolic pressure between 90 and 120 millimeters of mercury, and diastolic pressure between 60 and 80. Taken together, a normal reading is a bit less than 120/80. The blood traverses the landscape of the body through the pipes of the circulatory system. In any plumbing system, several things can increase the force on the walls of the pipes: the properties of the fluid, extra fluid, or narrower pipes. So if the blood thickens, a higher pressure is needed to push it, so the heart will pump harder. A high salt diet will lead to a similar result. The salt promotes water retention, and the extra fluid increases the blood volume and blood pressure. And stress, like the fight or flight response, releases hormones like epinephrine and norepinephrine that constrict key vessels, increasing the resistance to flow and raising the pressure upstream.
On purpose.
Blood vessels can usually handle these fluctuations easily. Elastic fibers embedded in their walls make them resilient. But if your blood pressure regularly rises above about 140/90, what we call hypertension, and stays there, it can cause serious problems. That's because the extra strain on the arterial wall can produce small tears. When the injured tissue swells up, substances that respond to the inflammation, like white blood cells, collect around the tears. Fat and cholesterol floating in the blood latch on too, eventually building up to form a plaque that stiffens and thickens the inner arterial wall. This condition is called atherosclerosis, and it can have dangerous consequences. If the plaque ruptures, a blood clot forms on top of the tear, clogging the already narrowed pipe. If the clot is big enough, it can completely block the flow of oxygen and nutrients to cells downstream. In vessels that feed the heart, that will cause a heart attack when oxygen-deprived cardiac muscle cells start to die. If the clot cuts off blood flow to the brain, it causes a stroke. Dangerously clogged blood vessels can be widened by a procedure called an angioplasty. There, doctors thread a wire through the vessel to the obstructed site, and then pass a deflated balloon catheter over the wire. When the balloon is inflated, it forces the passageway open again. Sometimes a rigid tube called a stent is placed in the vessel to help hold it open, letting the blood flow freely to replenish the oxygen-starved cells downstream. Staying flexible under pressure is a tough job for arteries. The fluid they pump is composed of substances that can get sticky and clog them. And your typical healthy heart beats about 70 times a minute, and at least 2.5 billion times during an average lifetime. That may sound like an insurmountable amount of pressure, but don't worry. Your arteries are well suited for the challenge.
All right, there we go. Thank you. Next week we'll have our quiz and our lab dissecting a heart.
My grandma had to get 2